Okay, you guys, I happened to get lucky and grab a really good slide first off. This is the one that's labeled liver of pig. It's an old slide. The label's really, really faded. There are some newer ones that stain pretty much a uniform pale pink and can be a real challenge to try and get a handle on. This one is stained in such a way that you can see the connective tissue elements between what are called liver lobules. And this is the connective tissue of what I speak, separating the liver into lobules. So this would be one lobule, this would be another one, this would be another one. And the lobules tend to have relatively regular straight edges. They look like polygons, usually. Somewhere close to the center of each lobule is a central vein, right here. But it's out here in these connective tissues where the rest of the stuff that we're interested in is typically found. And specifically, we're going to look for a cluster of three structures called the hepatic portal vein, the hepatic artery, and the hepatic duct. Those three things make up what's called a portal triad or a portal tract, depending on the book that you happen to be looking at. The easiest place to find those three things are where connective tissues from different lobules merge. So this area would be a good area to look for a portal triad. Probably this area would too. And probably there's a junction down here as well. Now if you have one of the pale slides, it can be really difficult to locate those connective tissues that are separating lobules. However, the central veins usually are nice and open like that. You can see a central vein, know that you're going to have to move away from it, and that you should hit a connective tissue not too far away. Once you find that connective tissue, track it until it forms a junction or a union with the connective tissue of other lobules. Now, let's zoom in on this one and see what we can see. It's pretty small, but maybe we'll get lucky. Right there is where I'm going to be looking. It's not looking promising. Yeah, not good. Not good. There is one structure here that's kind of occluded in the lumen. It appears to be a branch of the hepatic portal vein, but because you guys haven't seen these liver slides before, it's difficult for you to tell. Right here, branch of the hepatic portal vein, I can tell that because this is a squamous epithelium right here. If this had been the branch of the hepatic artery, there would be a thin but distinguishable band of smooth muscle surrounding it. We don't have that here. If it had been a branch of the duct, it would be lined with cuboidal epithelium, which we don't have here either. Now, ideally, we should see all three things right in this area. I'm only seeing one that happens.
I have it if you want my slide.
You got a good one?
Yeah.
Let me scan around in this one for another minute or so and see if we can find something better. If not, I'll look at your slide. So I backed out to low power and at that junction I can see that there's a large hollow tubular structure, a very large one, but I'm going to zoom in on that connective tissue next to it to see if I can see the other structures. The size will vary between these three structures, whether you've got a duct, an artery, a vein, or a combination of them. It appears as though that large tubular structure right there is a blood vessel, although I'm going to have to zoom in to check. This is a poor example, though. This apparently is not going to be very good for locating portal triads. It's certainly good for identifying the lobules, though.
normally would all three of the vessels be roughly the same size, or
No.
will one generally be bigger, or like?
It will vary, but generally the hepatic portal vein is the largest structure there. And the remaining two, the duct and the artery, are usually about the same size, although that can vary too. Now this is just a general rule of thumb. A lot of exceptions are there.
Sure.
I've got two of the three structures here. I've got a wide branch right here of the hepatic portal vein, the squamous epithelium. In the lower right-hand corner, a little tiny tubular structure with cuboidal epithelium is the duct. So there's the hepatic duct right there. And this great big thing is a branch of the hepatic portal vein. You can see it's branching. So this slide is not very good for liver. I'll go ahead and give yours a try, Jamie. Some of these slides are really good, some of them are really poor. So just like everything, it's luck of the draw. So I can see faintly the connective tissue elements already. They're kind of subtle, but you can see right in the center of the screen there's a junction where connective tissue elements are coming in from three directions. That possibly could be a good place to look, although actually I can see some right here under low power. So I'm going to zoom in on this area. Must have gone there.
I'm so happy about when I
Okay. Now you're not going to be real happy with this, but I've got two of the three elements here. Here's the duct. You can make out the big nuclei, how close they are together. You can see that based on the position of the nuclei, that the cells are roughly as tall as they are wide. So this is a cuboidal epithelium. This is the duct right here. This and this are both blood vessels. And based on the thinness of their wall, they both appear to be hepatic portal vein. If they were artery, they would have an obvious band of smooth muscle around them. So we've got two of the three elements here.
Jamie, I think I have it pretty good here, and it's blue. It's the older slide. It's got a nice structure.
Let me scan around a little bit more here and see if I can find another good one. Yeah, I might have all three right there. Ha ha ha! There's an artery right in the middle of the field of view. So it's subtle, but see the smooth muscle band right there? Going around it?
Cool.
That's an artery, hepatic artery.
Yeah, where did I have a science?
Right there, kind of hidden, oval, is the hepatic duct.
Oh.
You see the epithelium?
Mm-hmm.
Subtle, but it's there. That's cuboidal. And yeah, it looks like we have kind of a weird vein right here this appears to be connective tissue right here. Imagine a blood vessel that's just slightly lifting out of the plane and then diving back in. And imagine that we're cutting through the tunica externa in portions of that. I think that's what this is right here. And then we're seeing a bit of lumen, and it's rising out, and it's diving back down right here. This is lumen as well. So that should be vein. hepatic portal vein, although it's not a good one. But that artery down there, that's textbook perfect right there. Yeah, I think both of those are hepatic artery as well. Looks like there is a bit of smooth muscle around them. Although not as good as the other one. So you can spend a lot of time on some of these slides looking for good examples of each, but if you just keep in mind that if you see something with cuboidal epithelium, it has to be duct. There's a good duct and an artery side by side right there. See the duct? Cuboidal epithelium? This is a little tiny artery, just a smidgen of smooth muscle around it. I bet this is portal vein. Yeah. There's another small artery right there too. So the lumen of the portal vein has some junk in it. Some red blood cells and also some stain and some debris and stuff. But this is the hepatic portal vein. This is another branch of hepatic artery. You see the thin band of smooth muscle around it. And then over here we have that duct. So there's all three right there. It took a while, but if you keep after it, there. You can see all three in the field of view now. It may take a while, but eventually you'll spot all three in the same region. Did I miss anything on this slide, you guys?
Sinusoid.
Oh, sinusoids. Sinusoids are the little channels.
Oh, and hepatocytes.
Yeah. So all this stuff right here, those are all hepatocytes. Literally liver cells. The little channels, these little white spaces, like this, and then running through here. This is probably one right here. This is one right here. Those are your sinusoids. That's where the blood is. And remember, from your blood tracing, it's at the level of the sinusoids where hepatic portal blood mixes with hepatic artery blood, which is very unusual. And then the blood passes through those sinusoids to the center of the lobule to a central vein. And that's what we started out with. So if I back down, there's the portal triad we were looking at. I'm at medium power. Yeah, that thing right in the middle of the field of view is a central vein. It's not a good one. Usually they're nice and round, but all the little white flecks in the liver tissue around that, those are all sinusoids, and they all converge on that central vein. Did I miss anything else, you guys? So, the quality of the slides is highly variable. Look at a number of them. All right, what's next? Gallbladder? Gallbladder.